Hello, good morning everyone. I myself Dr. D. Swapnasri, Assistant Professor in Botany from KVR Government College Women Autonomous Kurnool, Andhra Pradesh. Today I am going to present before you my research article which was published in ijsr with the title Talc-based formulations from potential biocontrol agents against yellow mold in groundnut Arachis hypoxia. Coming to the abstract, first step is isolation. Isolation of the rhizosphere microflora from the groundnut fields was done by serial dilution method, and the microorganisms isolated were screened for antagonistic effect against Aspergillus flavus. Two fungal strains, namely Penicillium species and Trichoderma asperellum, and four bacterial strains, namely Pseudomonas fluorescens, Bacillus megatiarum, Bacillus subtilis, and Bacillus subspecies, were isolated and studied through morphological and biochemical characters from rhizosphere of groundnut. Similarly, for seeds also we have done. Trichoderma asperellum among fungal isolate and Pseudomonas fluorescens from bacterial isolate showed higher antagonistic effect against Aspergillus flavus than other fungus and bacteria isolated from the rhizosphere, and thus proved to be potent biocontrol agents. Two talc-based formulations, one from Trichoderma asperellum and another from Pseudomonas fluorescens, were developed as an integrated disease management package against yellow mold caused by Aspergillus flavus in groundnut. Trichoderma asperellum was multiplied in vitro in molasses yeast medium, and the biomass along with the medium was incorporated into the talc. The talc was packed and stored in polythene bags. Similarly, Pseudomonas fluorescens was multiplied in nutrient broth and formulated with talc-based product. This was packed and stored in polythene bags. Coming to the introduction, groundnut, we all know that groundnut belongs to family Leguminosae and subfamily Papilionoidea. Its botanical name is Arachis hypoxia. The genus Arachis is morphologically well defined and clearly delineated from its closest relatives by the presence of geocarpet peg. The flowers are borne on the axils of the leaves and on primary and secondary branches. The groundnut, being a highly nutritive crop, is prone to attack by numerous diseases. More than 55 pathogens, including viruses, affect this crop, causing a great loss in the yield. Of the seed-borne and soil-borne diseases, collar stem, stem rot, and dry rot caused in the root, severe seedling mortality. It causes dry root rot, and all these diseases cause severe seedling mortality, resulting in the patchy crop stand and reduce the yield from 25 to 40%. Apart from this, aflatoxin is one of the major problems produced in the infected groundnut seeds by Aspergillus flavus and Aspergillus parasiticus at the end of the season under drought conditions as stated by Diner et al. Coming to the pathogen, Aspergillus flavus causes yellow mold or alpha root disease in groundnut. Seeds and unemerged seedlings attacked by the fungus are rapidly reduced to a shriveled, dried, brown or black mass covered by yellowish spores. The diseased plants are stunted and are often chlorotic. The leaflets are reduced in size with pointed tips and often show vein clearing. If affected plants are seen, the radicle is found to be lacking in the secondary growth or development. Coming to the materials and methods, here the work was carried out in 5 major steps: isolation of microflora, identification of biocontrol agents, characterization tests of biocontrol agents, screening of antagonistic effect against the pathogen, and talc-based formulation. So let us see one by one. Isolation of microflora. This was done by using direct play technique as given by Aneja in 2003. The method here involved is groundnut seeds were obtained from farmers for isolation of seed microflora. The seeds were surface sterilized with 1% sodium hypochlorite for 3 minutes. Then the seeds were rinsed 3 times with sterile distilled water to remove the disinfectant, dried on sterile paper towels, and plated on selective medium like King's B medium and PDA, supplemented with streptomycin sulfate for growth of bacteria and fungi respectively. The nutrient agar containing plates were incubated at 30 degrees centigrade for 48 hours for bacterial growth, and PDA containing plates were incubated at 27 degrees centigrade for 7 days for fungal growth. After incubation, colonies were picked from dilution plates and maintained as pure cultures in their respective agar slants with a periodic transfer from one medium to another medium for further studies. Coming to the second step, identification of biocontrol agents. Identification was done based on cultural and morphological characters of pure cultures of the isolates. The visual and microscopic examinations were done to determine the genus, and later identification keys and illustrated manuals were used for species separation. For fungal isolates, microslides of each culture were prepared in lactophenol cotton blue, examined under the microscope, observed their morphological characters, and identified with the help of keys provided by Ellis, Booth, and Subramaniam. The measurements of the vegetative structures were taken with the help of ocular micrometer. Coming to the third step, characterization tests of biocontrol agents. HCN production and production of siderophores are the two tests done for bacteria. HCN production was assessed by growing the bacteria in 10% tryptic soy agar or Pseudomonas agar Kingsby medium supplemented with glycine. Filter paper soaked in picric acid and sodium carbonate solution was fixed to the underside of the lids of the plates and incubated for 5 days at 30 degrees centigrade. 1 ml of the test culture organism is inoculated in nutrient agar plate except one, leaving one, that is as control. The plates are incubated for 24 hours at 28 degrees centigrade. Then Whatman filter paper impregnated with alkaline sulfate acid. If we observe a color change from yellow to orange, this indicates hydrogen cyanide production by the isolates. Next one is siderophore production. CAS method is used to determine siderophore production by soil rhizosphere isolates. An orange-yellow color indicates siderophore production and a blue color indicates no siderophore production. Coming to the fourth step, screening of antagonistic effect against the pathogen. All the fungal and bacterial isolates obtained from rhizosphere samples were initially screened by dual culture technique for antagonism in vitro against pathogen. 20 ml of prepared PDA was poured into Petri plates and allowed to solidify. After solidification of PDA medium, 5 mm diameter mycelial disc from the margins of 7-day-old culture of the pathogen and the biocontrol agents were placed at the opposite ends of the plate at equidistant and observed from periphery on the same day. The plates were incubated at 28 degrees centigrade and checked daily until the fungal growth on the control plate reached the edge of the plate. 3 replications were maintained in each treatment, and for each replication 3 plates were used. The in vitro assay was repeated twice to shortlist the biocontrol agent. Based on the in vitro screening, the potential isolate was selected and maintained on PDA slants for further studies. Coming to the major and the last step of the research work, tag-based formulation using Trichoderma species. It was grown in molasses yeast medium and for 10 days at 27 plus or minus 1 degree centigrade. Subsequently, broth cultures were homogenized using a mixer grinder. The homogenized liquid cultures were formulated using talc as a carrier material, and for talc we have used liquid broth culture of Trichoderma species in 2:1 weight by volume with 10 grams of carboxymethyl cellulose per kilogram of carrier material, which is used as adhesive. The moisture content was maintained and the population of Trichoderma was counted immediately after preparation. Similarly, the same procedure is followed for Pseudomonas fluorescens, but it was grown in nutrient broth medium and incubated at 28 plus or minus 1 degree centigrade for 48 hours in orbital shaking incubator at 100 rpm. After incubation, the broth cultures were formulated using talc as a carrier material in 2:1 ratio, and the moisture content was maintained and population was measured soon after preparation. So these are the materials and methods which we have carried out in 5 steps. Coming to the results and discussion, the fungi and bacteria associated with rhizosphere soil were isolated. From these 2 fungal strains and 3 bacterial strains were obtained. All the isolates were identified based on their morphological characters. The most prominent bacterial isolates were obtained from rhizosphere soils, and strains were studied for morphology by using Burgess Manual of Determinative Bacteriology. The shape, color, and type of the colony were estimated on the nutrient media, and further gram staining was also performed. In addition to these, the morphological and biochemical characters of these isolates were also tested, and the results were shown in table 1. So here if you see the characteristics, colony configuration, colony margin, surface of colony, pigment, gram reaction, and here spore formation, motility, cell shape, hydrogen sulfide production, and IMVIC test, and along with nitrate reduction, catalase test, oxidase test, and urease test were also carried out for identifying these bacterial strains. In addition to that, characterization tests were done for the main biocontrol agent, which is Pseudomonas fluorescens. Among the bacterial isolates, the cyanogen activity was present only in Pseudomonas fluorescens. After incubation, the color of the filter paper was changed from yellow to orange color. A change in filter paper color from yellow to orange brown was considered to be an indication of HCN production as stated by Donate Korea et al. In 2005. Microbial production of HCN has been suggested to be an important antifungal feature to control the pathogen. Cyanide acts as a general metabolic inhibitor to avoid predation and competition. If you see the siderophore production, which was carried out by CAS agar plate method, among the bacterial isolates, the siderophore production was observed only in Pseudomonas fluorescens. On CAS agar plate, an orange colored halogen, which you can observe in this image, an orange colored halogen around the bacterial colony observed was considered as an indication of siderophore production. And the 2 fungal strains which were identified by their morphology are found to be Penicillium species and Trichoderma asperellum. This is Penicillium species and this is Trichoderma species. And these 2 species were identified by their characteristic features. So Penicillium, the characteristic features of Penicillium being colonies of fungus grow well to a diameter of 5 to 6 cm in 7 days as dull green colored powdery mass. The reverse of the colony appeared purple-red color. Conidiophores arise from substratum smooth, variable in length, up to 200 micrometer long and 2.3 to 3.3 micrometer wide, biverticillate and symmetrical. Medullae were 5 to 10, measured of 9.3 to 14 into 2.3 into to 1.3 micrometer in size. Sterigmata were lanceolate and varied in size. Conidia were smooth, variable in dimensions, elliptical or globose, and 3 to 4.5 into 2.3 to 3.1 micrometer in size. By observing all these characters, we have identified that fungal strain as Penicillium. Similarly, for Trichoderma asperellum, the fungus grows rapidly with immense vegetative mycelium. Colonies were white at first, later turned to light green to deep green due to conidial mass. Conidiophores were distinct from vegetative hyphae, indefinite in length and dichotomously or trichotomously branched. Conidia were born in groups, green, smooth, thick-walled, globose or ovoid, 3 to 4 micrometer in diameter or 3 to 5 micrometer in size. Coming to the next step, screening of antagonistic effect against the pathogen. All the fungal and bacterial isolates obtained from the rhizosphere sample were screened by dual culture technique for antagonism in vitro against the pathogen. In vitro studies on dual culture in agar plate against Aspergillus flavus revealed that Trichoderma asperellum inhibited maximum followed by Pseudomonas fluorescens. Of the 4 bacterial strains and 2 fungal strains, Trichoderma from fungal strain and Pseudomonas fluorescens from bacterial strain were identified, and they to inhibit more against the Aspergillus flavus. Pseudomonas fluorescens shows inhibitory activity against Aspergillus flavus. The inhibition is thought to be because of production of secondary metabolites or some antimicrobial compounds as stated by Anjaya et al. In 2005. Coming to the tag-based formulation of Trichoderma aspergillum, which was found to inhibit Aspergillus flavus. It was grown in vitro in the laboratory. It was multiplied in molasses yeast medium for 10 days. Biomass along with the medium was incorporated into the tag, and the tag was prepared with 10 gram of carboxymethyl cellulose per kilogram of carrier material as adhesive. The moisture content was 8% and population was estimated as 2 into 10 to the power 7 immediately after preparation. The formulated product was packed and stored in polythene bags. So this was done for both Trichoderma aspergillum and Pseudomonas fluorescens. These are some of the references which I have followed in my research work. Finally, I would like to thank my UGC who has funded me to carry out this work, and also to the principal of my college for providing facilities to carry out my project. I am very much thankful to IJSR journal coordinators for their motivation to submit my research work as e-presentation. Do you have any questions? You can feel free to mail to my mail ID ssdande750@gmail.com. Thank you.